Hello everyone, welcome to this new series of NEET revision flashcard questions. Today we are going to see some questions on biomolecules, which is NCERT chapter 9, which are NEET level questions. So first question is, which element is found in the highest proportion in the body? So hydrogen, carbon, oxygen and nitrogen. Which is the highest founding element within the body? So answer is oxygen. So oxygen is what it makes up 65% of the human body weight, and it's primarily due to the presence of water and organic molecules. Okay, next question. The acid insoluble fraction, remember this, acid insoluble fraction of a living tissue contains micromolecules, macromolecules, both micromolecules and macromolecules, and D, inorganic salts. What could be the answer? B, macromolecules. Why? Because macromolecules consist of proteins, nucleic acids, polysaccharides, which are all insoluble in the acid. So these are found in the acid insoluble fraction. Next question. Lipids, though having low molecular weight, are part of the acid insoluble pool. Why? They are hydrophilic, they form aggregates with membranes, they are covalently bonded, they are proteins. Okay, of course they are not proteins, so this option rules out. Okay, they are not hydrophilic also, they could be hydrophobic because they are lipids. So what could be the reason? They form aggregates with the membranes. Okay, so lipids associate with the membranes, making them part of the acid insoluble pool. So because of that, though lipids are low molecular weight compounds, they are found in the acid insoluble fraction. Which biomolecule has a Zwitter ionic form? Zwitter ion means both positive and negative charge. Okay, lipids, nucleic acids, amino acids, polysaccharides. Okay, so of course this term is related to amino acids. Amino acids can exist as Zwitter ions in solutions when both positive and negative charges. The bond between two amino acids is called glycosidic bond. No, it is happens within the carbohydrates. Peptide bond, yes. Phosphodiester bond, no, in the nucleotides. And hydrogen bond. So peptide bond is the correct answer. Proteins are polypeptides and they are linked by peptide bonds. Collagen is classified as an enzyme. No, it's not an enzyme. It is a protein rather. It is not a hormone. Okay. Structural protein and receptor protein. So it is basically a structural protein. It helps maintaining the structure within the tissue, and receptor protein is different thing. Receptor protein basically helps in signal transaction, so it is not a receptor protein. So collagen provides structural support in connective tissues. The primary structure of a protein refers to, remember we are talking about the primary, folding into helices. No, this is not. This is the tertiary structure. Okay, so this is the tertiary structure or the 3-degree structure. Linear sequence of amino acid, yes. 3D arrangement. This is the quaternary structure and subunit assembly. Okay, so option B is correct. The enzyme ribulose bisphosphate carboxylase oxygenase Rubisco is the most abundant enzyme on earth, found in animals, a storage protein composed of carbohydrates. So it is not found in animals, it is found in plants. It is not a storage protein. Okay, it helps in photosynthesis. It is not composed of carbohydrate, it is an enzyme. Okay, enzymes are composed of proteins, so it is the most abundant enzyme on earth, and it is involved in the initial carbon fixation step of photosynthesis. Okay, which of the following is a disaccharide? Glucose, of course it's not, it's a monosaccharide. Fructose, it is also a monosaccharide. Okay, sucrose is a disaccharide. Glycogen, no, it is a polysaccharide. So sucrose is formed by the condensation of glucose and fructose. Okay, so this is very important. The subunits of sucrose are glucose and fructose. The storage form of carbohydrate in animals. Cellulose, no, it is primarily found in plants. Glycogen, yes. Starch, it is also found in plants. And fructose, no. Okay. So it is glycogen is a preliminary storage polysaccharide and it is found mostly in the liver and the muscles. So which carbohydrate is not a homopolymer? Okay, so this refers to not a homopolymer. Starch, glycogen, cellulose, chitin. So chitin is not a homopolymer, it is rather a heteropolymer made up of modified sugars of N-acetyl glucosamine. Okay. Cellulose cannot be digested by humans because it lacks glycosidic bonds. Humans lack cellulase. It is water insoluble. It is a branch structure. So humans lack cellulase and cellulase is important for degrading cellulose because it has beta 1,4 glycosidic bonds. Which of the following is a phospholipid? Okay. Lecithin, cholesterol, palmitic acid, arachidonic acid. So lecithin is a phospholipid and it is primarily found in cell membranes. The presence of double bonds in a fatty acid makes it, what do you think it makes it? It makes it saturated, unsaturated, hydrophobic, or amphipathic. Unsaturated means you still have presence of free groups. Okay, saturated means completely they are saturated with all the valence positions of carbons are occupied. Okay, so if all the valence positions are occupied, you will have only the single bonds, and unsaturated means if you have double bonds, those double bonds could be opened and occupied by another group. So the correct answer is unsaturated fatty acids. So which lipid forms the backbone of cell membranes? Backbone of cell membranes is formed by phospholipids because they form the bilayer structure of the cell membranes. Okay, so this is correct. Which sugar is present in RNA? Ribose, deoxyribose, fructose, glucose. Ribose is the correct answer. Deoxyribose, no. It is present in DNA. Fructose, no. Glucose, no. Okay. So the backbone of DNA. Remember, we are talking about the backbone. Is made up of nitrogen bases, deoxyribose and phosphate groups, amino acids, fatty acids. So deoxyribose and phosphate groups are what which support the nitrogen bases. So they are called as the backbones. Which nitrogen base is not found in RNA? Adenine, thymine, uracil, guanine. Adenine yes, thymine no, uracil yes, and guanine yes. So correct answer is thymine. Nucleotides are composed of amino acids, sugars, and phosphates. Nitrogen bases, sugars, and phosphates. Lipids, sugars, and nitrogen bases. Sugars, lipids, and phosphates. So the correct answer is nitrogen bases, sugar, and phosphates. Okay. Enzymes lower the activation energy of the reactions by breaking bonds, providing an alternative pathway, increasing substrate concentration, raising temperature. Okay. So of course they don't make break bonds, they provide an alternate pathway. They don't increase the substrate concentration, they can't increase rather substrate concentration, and they usually do not raise the temperature. Which enzyme converts glucose to pyruvate in glycolysis? Glucose to pyruvate. Okay, which enzyme they say? Hexokinase, Rubisco, carbonic anhydrase, amylase. So option A is hexokinase. Okay, actually the more specific question should be glucose to glucose 6 phosphate. This is what hexokinase does, not glucose to entire pyruvate, because glycolysis is played by many enzymes. Hexokinase is one of them. Okay, it is not like it is directly converting glucose to pyruvate, but it is one of the important enzymes in the glycolysis step. Okay, this question could be more specific. Ribozymes are protein enzymes, no. Protein RNA molecules with catalytic activity, yes. That is why they are called as ribozymes. DNA molecules, carbohydrate polymers. Okay. So they are ribozymes, RNA molecules that function as enzymes. What is the active site of an enzyme? A substrate binding site, a site for competitive inhibition, the coenzyme attachment site, or a non-functional domain. So the answer is a substrate binding site, which is called as active site and facilitates the catalysis. Okay. Which biomolecule is amphipathic? Okay. Amphipathic means it has tendency towards both. It could be both hydrophilic or non-hydrophilic. Okay. So the correct answer is phospholipid. Okay. Hydrophobic and non-hydrophobic regions. Chitin is a protein, polysaccharide, lipid, nucleic acid. So of course it is a polysaccharide. Which of the following organic compounds is a polymer of glucose? Okay, glycogen, lecithin, arachidonic acid, adenine. Glycogen is a polymer of glucose. Lecithin is a phospholipid. Arachidonic acid is a fatty acid, and adenine is a nitrogen base. Okay, so the answer is glycogen. So the molecular weight range of micromolecules, here we are talking about micromolecules, is approximately less than 1000 daltons, 1000 to 100,000 daltons, greater than 100,000 daltons, and 5000 to 20,000 daltons. So it is less than 1000 daltons. Those are micromolecules. Which of the following is not a primary metabolite? Amino acid, sugars, alkaloids, fatty acid. Amino acids, yes. Sugars, yes. Alkaloids, no. These are the secondary metabolic products. Fatty acids, yes. Okay. And it is mainly plants, because why they are called secondary metabolites? Because they have no direct role in primary physiological processes. Which class of biomolecules includes lecithin? This we have already discussed. Polysaccharides, proteins, phospholipids, nucleic acids. So they are phospholipids. The unique chemical property of amino acids is due to the presence of amino group, carboxylic group, R group, hydroxyl group. So it's basically this R group which confers a unique chemical property to each and every amino acid, because this R group changes with the change in amino acid. Okay, the rest of the groups remain the same. Which of the following amino acid is aromatic? This is also important question. Serine, no. Valine, no. Phenylalanine, yes. Glycine, no. The correct option is phenylalanine, because it is an aromatic. Other two amino acids which are also aromatic are tyrosine and tryptophan. The 3D folding of a polypeptide into a functional protein is called primary structure, secondary structure, tertiary structure, quaternary structure. So it is the tertiary structure. It's the overall 3D folding of a single polypeptide. And a quaternary structure is when two or more functional polypeptide chains come together, then they form the quaternary structure. What type of bond stabilizes the secondary structure of proteins? Here we are talking about the secondary structure. Covalent bond, hydrogen bond, ionic bond, phosphodiester bond. Answer is hydrogen bond. Okay. It is primarily found in secondary structures like alpha helices and the beta pleated sheets in proteins. Okay. Which protein is most abundant in the biosphere? Okay, most abundant protein in the biosphere. Keratin, rubisco, myosin, or collagen? Answer is rubisco. Okay, because it is found in plants, and plants, there are much more number of plants are there in animals. So it is the most abundant protein on earth. Which of the following is an example of a storage polysaccharide? Okay, chitin, glycogen, cellulose, inulin. This we have already discussed earlier. Glycogen is a storage polysaccharide in animals, in the liver and muscles. The helical structure of starch is responsible for binding with lipids, proteins, iodine, water. Okay, so the answer is iodine. So there is a test for presence of starch which we do with iodine. and the color that we get is a blue color. It is due to the complex of binding of iodine with starch due to its helical structure. The reducing and non-reducing ends of a polysaccharide refer to sites of branching, free aldehydic or ketonic groups, bonding of amino acids, phosphate groups. So, free aldehydic or ketonic groups. So, if one end has a free aldehydic or ketonic group, that end is called as a reducing end. Okay, and if the end does not have that, then it is called as a non-reducing end. Why? Because these can get oxidized, aldehyde, this end can get oxidized and in turn reduce the other compound. Okay, so in oxidation reduction reaction, in a redox reaction, one thing gets oxidized, another thing gets reduced. So if you have a free end, okay, a reducing end, which is a free aldehydic or ketonic group, they can get oxidized to aldonic acids, like glucose can get oxidized to gluconic acids, and in turn it can reduce the other compound. So because this end can reduce the other compound, so this is called as a reducing end. While if you do not have this free aldehydic or ketonic group, then it is called as a non-reducing end. As fatty acid with no double bonds, okay, you don't have any double bonds, is called saturated fatty acids. Okay, which of the following is the component of cell membranes? Cholesterol, triglycerides, glycogen, cellulose. So cholesterol is a component of cell membranes. It is a lipid that stabilizes the fluidity of the cell membranes. So this is a very important function of cholesterol. Which lipid contains glycerol as its backbone? Cholesterol, phospholipids, palmitic acid, steroids. So phospholipids are composed of glycerol, fatty acids, and a phosphate group. Okay. Which of the following is a nucleotide? Adenosine, adenine, ATP, deoxyribose. Adenosine is a nucleoside. Adenine is a nitrogen base. ATP is a nucleotide. Deoxyribose is a sugar. The sugar present in deoxyribonucleic acid is glucose, ribose, deoxyribose, fructose. So as you can make out from the name, it is deoxyribose. The pyrimidine bases found in DNA are adenine and guanine, cytosine and thymine, guanine and cytosine, uracil and adenine. Okay. Remember, we are not talking about purines here, we are talking about pyrimidines only, and that too in DNA. Okay, what will be the answer? Cytosine and thymine. Enzymes increase the rate of the chemical reaction by consuming reactants? Of course not. Lowering the activation energy? Yes. Increasing the product stability? No. Binding irreversibly to the substrate? No. Because enzymes are like replenished after the reaction. The active site of an enzyme is rigid and fixed, highly specific to the substrate, composed of DNA, non-functional in the tertiary structure. So the correct answer is highly specific to the substrate. It is a site with the substrate binds and it is specific for each substrate. It is not rigid and fixed, although it is not composed of DNA, enzymes or proteins, and it is not non-functional in the tertiary structure. Otherwise it wouldn't have bind to the substrate. Which factor does not affect enzyme activity? Temperature, substrate concentration, inhibitor presence, DNA sequence. Temperature, of course it affects. Substrate concentration, of course it affects. Inhibitor presence also affects. It lowers the enzyme activity. And DNA sequence. There is no topic of DNA sequence here. Competitive inhibitors bind to the substrate, the active site of the enzyme, product and coenzymes. So competitive inhibitors compete with the substrate to bind to the active site of the enzyme. The dry weight of living tissue mainly consists of inorganic salts, water, organic macromolecules, micromolecules. So dry weight consists of organic macromolecules like proteins, nucleic acids, polysaccharides. These three biomolecules compose of most of the dry weight. The active site of an enzyme is rigid and fixed, highly specific. This we have already discussed, composed of DNA, non-functional tertiary structure. So it is highly specific to the substrate. Which of the following are secondary metabolites. Okay, same some of same question which we had discussed: proteins and nucleic acids, alkaloids and flavonoids, glucose and fatty acids, water and ions. So alkaloids and flavonoids are the secondary metabolic products in plants. Okay, so with this we come to today's flashcard questions on biomolecules. Because this channel is still growing and yet to be monetized, I would be grateful if you guys can contribute something, some amount as per your wish, to help in growth of this channel and support me, and like make help me to make more videos, help me to grow this channel and continue making more videos so that you guys can be benefited and educations could remain free.